Do you ever wonder how your body accomplishes all the things it does to keep you alive? Well, the answer to that question is almost always proteins. Today we explore the process through which proteins are made, known as RNA translation.
The first component of RNA translation is the mRNA, or messenger RNA. After being created in the nucleus through transcription, the mRNA comes out into the cytosol. The mRNA is a single helix strand of genetic information which contains nucleotides in sets of 3, known as codons. Each of these codons corresponds to a specific tRNA, which brings a specific amino acid to the ribosome to be attached to the polypeptide chain.
Here, a large ribosomal subunit and a small ribosomal subunit attached to the mRNA. Together, these form a ribosome. The large ribosomal subunit contains an A site, which binds to amino-acyl tRNA, a P site, which binds to peptidyl tRNA, and an E site, which binds a free tRNA before it exits the ribosome.
A tRNA with its corresponding amino acid attached comes into the A site, where its anticodon binds with the codon on the mRNA. This tRNA is then moved to the P site as a new tRNA enters the A site. The first tRNA's amino acid is detached and it is moved into the E site. The second tRNA then moves from the A site to the P site, where its amino acid is attached to the first amino acid, forming a polypeptide chain. This happens many times, translating the entire strand of mRNA, forming a long polypeptide chain that will become a protein.
DNA shotgunning is a form of DNA sequencing used by the Human Genome Project. After much research to find the most efficient way to sequence the human genome, DNA shotgunning was developed.
So DNA shotgunning is a type of DNA sequencing used by the Human Genome Project where the people take a long strand of DNA and they can shotgun it, so to speak, into a bunch of smaller pieces made up of 1 to 2,000 nucleotides.
So we explode a long molecule of DNA to about 1 billion pieces, and then we take the pieces and we put them into the computer in order to sequence the DNA.
And then all of these go into a single library of information from the original DNA molecule, and they are cloned so that the people working on the Human Genome Project can easier deal with, like, rather than one large strand, they can deal with a bunch of small ones.
But the fragments are very repetitive, so it doesn't work a lot of the time. So we fill in what we can, and what we can't, we fill in with the established public data.
When we started the Human Genome Project, our method was based on splitting the DNA molecules into 150,000 base pairs, which was called map-to-map or BAC sequencing.
The Human Genome Project started all off with the map sequencing, the BAC to BAC. But us at Celera, we took it up a notch with the DNA to the shotgunning sequencing, which took it down from about 150,000 to 1 to 2,000 base pair sequences.
Once Celera invented the shotgunning process, we realized it was a much more effective method for our project, and that's what we ended up finishing the Human Genome Project with.